Hello, everyone. No response. Anyways, I would continue. Because of, I am in the department and there is an issue of internet. So I could not connect the desktop, though I could connect you with my mobile. So till the last class, we had discussed how the immune system works. We have discussed about innate immunity, adaptive or the acquired immunity. So innate immunity consists of the WBCs, the different types of WBCs, which take part in giving us the defense, and the adaptive or acquired immunity consists of two types of cells, T and T cell, T lymphocyte, and B lymphocyte. The major function of B lymphocyte is to produce the antibodies, and the T lymphocyte, which are of two types, depending upon the type of cluster designated molecule is attached with their membrane. If it is CD4, we call it T4 lymphocyte. If it is CD8 molecule, which is attached to the membrane of the T cell, it will be called T8 lymphocyte. So TCD4 or TCD8, and the TCD4 has got an ability to, recognize the antigen and keep it for the further usage, further need. It memorizes, it keeps the antigen affinity, the antigen binding sites, and thus it will help in producing the antibodies by B cell. So there are, and apart from that, there is another set of molecules which we call a set of cells, which we call as what we call the APCs, antigen presenting cells. They are usually dendritic cells. There are many types of dendritic cells in different tissue levels. So they usually present the antigen which is foreign to the body, either to the cytotoxic or the CD8 T cell, or they might straight away present it to B cell to produce the antibody. So I think much of the time has been just wasted. So we will not go into the details today. But I would be like putting up the videos regarding the antigen antibody reaction. Now one thing you need to understand, and before understanding those topics, you must have heard about agglutination. Have you heard of agglutination? And you must have heard about the word precipitation too. So the agglutination and precipitation, there is a difference between these two. But what is the difference? Before that, we would like to know what exactly these antigen antibodies react with each other. As antigens and antibodies both are protein molecules, they have amino peptide sequences, they have amino acids in their chain. So what happens, after they get in an active form, there are sites at which, suppose if it's an antibody, if it is binding to an antigen, it has to have a site where it gets it bound. So binding takes place as per, you have already known how this lock and key method works. So with the help of those lock and key methodology only, the antigen and antibodies bind each other. The binding site in the antigen, that means, sorry, the portion to which the antibody binds, the portion of antigen to which an antibody binds is called epitope. Okay, and the portion of antibody to which an antigen binds is called paratope. So this is an interaction between the epitope and the paratope portions of antigen and antibody in the methodology of lock and key method. So antibodies usually have these paratopes in which the epitopes of the antigens are attached or bound. And then if the antibody is free enough, then the antigen and antibody which has bound together will precipitate. Okay, and if the antigen is on some cellular portion and the antibody binds to it, and then they make the cell body not functional, or they make the cell body slower in giving any response. So that is called agglutination. Like as in agglutination, जिसे हम कहते हैं coagulation भी एक word आपने सुना होगा. Coagulation is a type of agglutination only. What happens in that? If it is, we have different blood groups, right? A, B, O, and AB. That means if the A antigen is present on the RBC membrane, A type of protein, we call it as blood group A. And if the B type of protein is present on the membrane of the RBC, we call it as B blood group. If both are present, we call it AB group. And if both are not present, we call it O group. There is no antigen present. And what happens when, if at all, we give an antibody of that A protein, which is present on the membrane of the RBC, so the proteins will be, the protein molecule, the A protein, which is present on the membrane of the RBC, will be bound by the antibody of A. And what happens? They try to make the RBCs to get settled. They do not flow in the bloodstream. They get settled. And that settling of the cells with the help of antibodies binding to the antigen present on the membrane is called agglutination, or the coagulation is a different aspect altogether. This is called agglutination of the antigen A that is present on the surface of the RBC. And similarly, if the B protein is present and we have provided the antibody B, then those cells, those RBCs which have B protein, B type of antigen on the surface, will be agglutinized by the antibody B. That way, they are being, settled down. When it is a cellular thing which has been settled down, we call it agglutination, because the cells get agglutinated at one place. And if it is a molecular level, the compound level, then we call that particular antigen which is free in the blood. If it is being bound with an antibody against it, so it will get precipitated, as we see in our chemistry. One compound is precipitated with another compound. It's like that only. If a protein and antigen, if it is bound with an antibody, and if the protein is free, then the whole molecule, the complex molecule with antigen and antibody, will settle down. And that settling is called precipitation. Okay, now coagulation is a bit different. Coagulation is, clot. Coagulation leads to the clots. Clots means what? The protein which is present in the blood which is called fibrinogen. That fibrinogen, in presence of external oxygen, external atmospheric exposure, they do what? They get turned into fibrins, and this fibrins, that is a productive or a type of process by which the flow of blood is blocked. So the portion which has been hurt or cut, that portion in which the blood is flowing, the vessels, if that portion is cut or hurt, those portions are, they have been, blocked by this fibrin, which is being converted as fibrin with the help of fibrinogen, the protein which is present in the blood. So this will create a mesh sort of fiber structure, which would stick at the opening or the damage or the cut portion of the vessel, so that it will not allow the blood to flow out. It stops the bleeding. So that coagulation is a bit different aspect. Agglutination is that it's a type of precipitation, it's a type of settling down of the cells, or cells having that antigen, or simply the antigen which will flee in the blood. So that is precipitation and agglutination. I would give you the details of the clips by which you can understand these things. And as I told, the antibody and antigen, the epitopes and the paratopes, we need to understand these epitopes and paratopes. And there are 5 types of antibodies. You must have heard the major, the prime antibody is the antibody G, which is also called as immunoglobulin. Globulins are those protein molecules which are not straight chained. When a protein molecule is a straight chain, then they are usually called as albumins. And when these straight chains get into some structures, globular structures, they are called globulins. So these antibodies are the globular structures which are not straight chains in a single chain. So they have some their own structure because of the intermolecular amino acid reactions, interactions, and that's why they have a different structure apart from the long straight chain structure. So these structures, they have a different affinity to different molecules, and these antibodies, which are proteinaceous, they are not straight chains, single straight chains. They are modified straight chains, and they, they have got the ability to settle down the antigen or the antigen which is present on the cellular membrane. So with the help of epitopes and paratopes, they try to precipitate the interaction between them. And globulins. So these globulins are, they participate in the immune system. So we call it them as immunoglobulins. So the antibodies are also called as immunoglobulins. And the major type is of G, the immunoglobulin G. And there's another one which is called immunoglobulin A, immunoglobulin D, immunoglobulin E, and immunoglobulin M. So the only thing you need to remember is the major immunoglobulin which is there is called immunoglobulin G. Of course, there are subtypes of immunoglobulin, 1, 2, 3, 5. There are different subtypes of immunoglobulins. They all are classified under the immunoglobulin G category. G has got, there are a typical immunoglobulin has a structure of Y shape. That means two chains of molecules which are attached. जहाँ मेरी ये घड़ी है, सपोज करिए वहाँ से अटैच हो गए, तो एक प्रकार का इम्यूनोग्लोबुलिन बन जाएगा. दोनों चेन्स घड़ी वाली पोर्शन पे दोनों से अटैच हो गए. एंड देन दीज फनल शेप वाला जो पोर्शन है, उसके साथ में एक और इम्यूनोग्लोबुलिन अमीनो एसिड का चेन अटैच रहता है, जो ये फ्लैंक्स हैं, फ्लैंक्स के साथ में अटैच रहता है. तो डबल चेंज स्ट्रक्चर्स नजर आते हैं, यानी कि ये भी डबल चेन स्ट्रक्चर्स नजर आएंगे और यहाँ पे भी डबल चेन. लेकिन ये जैसे वॉच वाले एरिया पे मान लो अटैच है, तो वो टिपिकल एक इम्यूनोग्लोबुलिन का स्ट्रक्चर है. यू विल सी इन द क्लिप व्हिच आई विल अपलोड इन द स्टडी मटेरियल. और दूसरी बात ये है कि ये जो अटैचमेंट है जो है इन स्ट्रक्चर्स के बीच में, इन चेन्स के बीच में, वही उनको डिफरेंशिएट करता है जी डी ई में। सो जी और डी ई जो है वो यदि आप देखोगे उनमें तो वो नॉर्मल जहाँ पे मेरी घड़ी है उस जगह पे यदि बहुत सारे मॉलिक्यूल्स एक दूसरे से अटैच हो जाते हैं, यानी कि सिस्टीन सिस्टीन है तो बॉन्ड बन जाएगा उनके बीच में, यदि ऐसे बहुत सारे मॉलिक्यूल्स के साथ में वो अटैच रहेंगे तो हम उनको यूजुअली वो जी वाले होते हैं, जी इम्यूनोग्लोबुलिन्स होते हैं। डी में मेरे ख्याल से 2 या 3 बॉन्ड्स रहते हैं और ई में भी 2-3 बॉन्ड्स रहते हैं, लेकिन डी और ई में फर्क यहाँ पे आता है एंड ऑफ द जो वाई शेप का जो बेस है वहां पे कुछ अटैचमेंट्स रहते हैं, वो अटैचमेंट्स रहे तो वो डी कहलाता है और नहीं रहे तो वो शायद ई कहलाता है। एंड अब बचे ए और एम। जो इम्यूनोग्लोबुलिन ए है वो डायमेरिक है, डायमेरिक मतलब 2 इम्यूनोग्लोबुलिन्स एक साथ रहते हैं। यूजुअली देखने में वो टिपिकल डी मॉलिक्यूल जैसा ही, डी इम्यूनोग्लोबुलिन डी जैसे ही या जी जैसे ही नजर आते हैं। 2 Y shaped molecules attached from the base, Y के जो नीचे होते हैं एक स्पेशल प्रोटीन के थ्रू, which is through and through the जो नीचे का पोर्शन है Y के नीचे के पोर्शन से अटैच रहता है। You will see the structures and डायमेरिक होता है, यानी कि हमेशा 2 मॉलिक्यूल साथ में रहते हैं। वैसे ही इम्यूनोग्लोबुलिन M जो है वो 5 मॉलिक्यूल पेंटामेरिक, पेंटामेरिक मतलब 5 एक साथ फ्लावर शेप में 5 एक साथ ऐसे लगे रहते हैं with the help of one single protein जो उन 5 को जोड़ के रखता है। तो वो यूजुअली कहलाते हैं इम्यूनोग्लोबुलिन M। और सबसे ज्यादा बी सेल्स के ऊपर, बी सेल्स के सरफेस के ऊपर इम्यूनोग्लोबुलिन M रहते हैं, बिकॉज M जो है 5 मॉलिक्यूल्स हैं इम्यूनोग्लोबुलिन के। हर इम्यूनोग्लोबुलिन के पास 2 पोर्शन है बाय विच इट कैन बाइंड टू द एंटीजन, यानी यदि पेंटामेरिक रहा तो 10 मॉलिक्यूल्स को एक साथ वो अटैच हो सकता है, क्योंकि 5 रहे तो हर एक के पास 2 हैं तो 5 मॉलिक्यूल्स है तो 10 एंटीजन को रेस्पॉन्ड कर सकता है या इंटरैक्ट कर सकता है। तो दीज ऑल थिंग्स यू विल सी इन दोज क्लिप्स। और बाद सेल मीडिएटेड जो रेस्पॉन्स है, क्या कहना चाहिए, बी सेल इनिशिएटेड रेस्पॉन्स या फिर टी सेल इनिशिएटेड रेस्पॉन्स, हम एक बार फिर से एक बार उसको एक प्रोसेस को समझने की कोशिश करेंगे। एंड ये सब जो इनिशियल फंडामेंटल थिंग्स हैं, जो प्रिलिमिनरी डिटेल्स हैं, जो मैं आप लोगों को विद द हेल्प ऑफ क्लिप्स जो है मैं अपलोड कर दूंगा। एंड ऑफ कोर्स जब हम नेक्स्ट क्लास में मिलेंगे देन आई वुड प्ले आल्सो सो दैट यू विल, if you have any query so you can interact with me or you can question it to me. Anyways, आज के लिए बस इतनी। Because of the internet issue we could not start on time, though I started the class, but internet was not there in the department, so I had to I could not connect with the desktop application of this app. Anyways, आज के लिए बस इतना। कुछ चीजें आप लोग जो मैं आप लोगों को मैं अपलोड कर देता हूँ उनको जरा जस्ट गो थ्रू इट व्हेनेवर यू अटेंड अ क्लास, जस्ट गो थ्रू द क्लिप्स व्हिच आई अपलोड सो दैट योर कॉन्सेप्ट्स विल बी क्लियर। एंड अब ये जो प्रिलिमिनरी इंफॉर्मेशन है अबाउट द एंटीजन एंड द एंटीबॉडीज, उनके बारे में मैं आपको वीडियो क्लिप्स अपलोड कर दूंगा। सो दैट्स ऑल फॉर टुडे, वी विल साइन ऑफ टुडे, वी विल सी यू नेक्स्ट वीक।